In this series of videos, we are taking a deep dive into the development of antidepressants. While learning about the history of 3 drugs, we are exercising skills connected to scientific discovery. If you are a pre-med college student, an AP high schooler, or simply someone with some background in biology who's interested in learning about the science of antidepressants, this series is for you. Years of research in the science of learning have demonstrated that deep engagement is the best way to learn and retain information. So try not to just watch this video. Take down some notes, answer questions, engage in the activities, and truly be here. With that in mind, in today's video, we will recognize the role of serendipity in scientific discovery, review experimental findings leading up to the development of a drug, examine the interactions of a drug with the human body at the molecular level, critique a hypothesis-driven study design. Welcome to the 1950s. This is a very tense political time around the world, but it's also an exciting time for modern pharmacology. Traditionally, pharmacology operated at the level of symptoms. Drugs were tested by looking at the effectiveness a substance had in treating or mitigating a disease. As you can imagine, one of the limitations of this approach is that many things can go wrong between ingesting a chemical substance and observing an improvement of symptoms. At best, you would have a very broad treatment, and at worst, a series of unanticipated side effects. However, the development of the ligand-binding assay, inspired by the work of Carl Landsteiner on immunization in the 1940s, allowed scientists to measure the binding affinity of a given molecule to the target involved in a disease. This was revolutionary for pharmacology because now they could look at the effect of a drug at the molecular level. Based on what's known about a disease from basic science, you may identify a drug target, and that is a marker molecule, often a protein, involved in the process that leads up to a condition. Then you can use a ligand-binding assay to test how a set of small molecules can bind to your target molecule. Given how sensitive the function of a biological structure is, this should break your macromolecule and ultimately disrupt the entire pathway leading to a condition. It's in this context that Drs. Selikoff, Robertsack, and Ornstein decided to test a recently developed antibiotic to treat tuberculosis. Yes, you heard me, this is still a video about antidepressants, but the pathways of scientific discovery aren't always that linear. The patients at Staten Island Seaview Sanatorium were given Iproniazid, a drug developed by the Hoffman-La Roche labs in 1952. Let's take a look at a 3D model of Iproniazid to see what we can learn just by looking at it. First, the basics. It has 9 carbons, 3 nitrogens, 1 oxygen, and 13 hydrogens. This amounts to a molecular weight of 179.22 grams per mole. So it's a small, rather light molecule. It has a formal charge of 0 because if you look at it, all the atoms in the structure are making the number of bonds they need to be stable. There are no positive or negative charges. But what about the distribution of charges in the molecule? This is a good moment to pause the video and try to identify what parts of this molecule are more electronegative. If you need a refresher on electronegativity, check out the link in the description. By now, you would have identified that this central part of the molecule is slightly more electronegative than the rest. That is, the negative charges are slightly more concentrated here because of the presence of our friends nitrogen and oxygen. Discussions about the distribution of electrical charges in the molecule are very important to drug development. Many considerations go into making sure that the drug can reach our drug target. There is often a tension here. To reach the target, our drug would have to cross many membranes, and for that, it needs to be nonpolar. At the same time, if the drug is too nonpolar, it can't be dissolved in water, and that also creates a problem for delivering the drug to the target. As you can imagine, finding just the right balance of properties can be challenging. This is a good time to talk about Lipinski's rules of 5. And I know we're still technically in the 50s, but let's skip ahead to the 1990s, when chemist Christopher Lipinski created a set of rules to determine the drug likeness of a molecule, based on physical-chemical properties observed in oral drugs approved at the time. For example, he found that most of them had a calculated oxygen-water partition coefficient below 5. This coefficient is determined experimentally, and it measures the polarity of a substance. Wouldn't you be surprised to see that Iproniazid is indeed inside this threshold? Lipinski's rules offer 4 heuristics to evaluate how likely it is that a small molecule will be an effective oral drug given its physical-chemical properties. Feel free to pause the video here and read an overview, but don't stress too much for now if you're a bit confused about them. We'll talk about each one in an upcoming video. Iproniazid fits all 4 of Lipinski's rules, and in physical-chemical terms, it's a quite predictable drug. But once we add biology to the equation, surprising things can happen. It all started when the doctors at Seaview Sanatorium made an interesting observation. Their patients were, and I quote, dancing in the halls though there were holes in their lungs. This is probably the funniest way someone could ever find of articulating that their patients were more euphoric than expected, especially given their clinical condition. This observation was about to change the landscape of treatments for depression. Until now, and remember, we are in the 50s, depression was perceived as a disease of the mind. Psychiatry was heavily influenced by Freudian psychoanalysis, and the idea was that psychotherapy was the only way to fix this mood disorder. For more severe cases, the standard treatment was electroconvulsive therapy, an unlocalized technique of applying electrical current to the brain in the hopes of changing its electrical activity and ultimately treat the symptoms of depression. Because depression and other psychiatric disorders were not seen as linked to a biological substrate, there were no chemical substances to target them. This is why the observation of euphoric footloose-like patients was so important. For the first time, there was a hint that a chemical substance could be impacting a patient's mood, something otherwise thought of as purely related to the mind. This ability to pay attention to details, even when they don't seem to have an immediate application, is recurring in science. If you've ever worked in a lab, you may have been introduced to lab notebooks. In there, researchers will write about anything, from the most crucial to the seemingly irrelevant observations. While many people tend to think of science as a very straightforward and rigid domain, a lot of scientific discovery happens just like that. While looking for something, as in this case they were looking for an effective treatment for tuberculosis, you may end up stumbling upon something else, like a potential treatment for depression. This is what we call a serendipitous discovery, or the discovery of something unsought, regardless of the systematic process that led to the accidental observation. This is not just a side element. Serendipitous discoveries contributed to the development of almost 1 in every 4 commercially available drugs. Back to our timeline, in 1957, Iproniazid was approved by the FDA to treat tuberculosis under the brand name of Marcelid. By then, the excitement about its psychic properties were rising. That's also when Lumor, Saunders, and Klein published the first paper about these properties, a clinical and pharmacodynamic evaluation of Iproniazid as a psychic energizer. They paved the way for a series of experiments about Iproniazid in the 1950s. But before we jump to the excitement, let's take a step back and think, what can we confidently say about Iproniazid? If you really think about it, the only thing we can say for sure is that Iproniazid can treat tuberculosis. After all, that was the end approved by the FDA. By this time, it was also known that Iproniazid could inhibit monoamine oxidase activity. Monoamine oxidases are a family of enzymes present in the outer membrane of mitochondria. We find them in many tissues in the body, including the central nervous system. They catalyze the oxidation of monoamines such as serotonin, dopamine, adrenaline, and noradrenaline. To summarize, monoamine oxidase decreases the availability of monoamines. Thus, as an inhibitor of this enzyme, Iproniazid ultimately increases the concentration of monoamines. Does that piece of information make you feel a bit more confident that Iproniazid could treat depression? I may have been too revealing here, but if not, don't worry, we'll get back to it. For our purposes, the natural next step would be to ask, can Iproniazid treat depression? Coming from this research question, how would you frame the hypothesis to test? I like to use the if-then framework because I think it covers all the 3 parts of a plausible and testable hypothesis. If we do X, then we expect to see Y because of Z. Try pausing the video here and writing down your hypothesis as an if-then-because statement. Once you have your first draft, hit play. Now look at each portion of your hypothesis and ask, Is this precise enough that someone else could design an experiment measuring the same independent variable? How would they know that we got the same or different observations for the dependent variable? Is this explanation enough to convince them we found a plausible mechanism? With this in mind, pause the video once more and try rewriting your hypothesis. Was there substantial improvement? Can you make it even more specific? When you think your hypothesis is good to go, hit the play. We are now looking at one of the studies that attempted to ask the same research question, but that might have had a different hypothesis than the one you proposed. It's a clinical and biochemical trial by Parry and Sandler conducted in 1959. Let's see a brief overview of the design. A sample of 50 patients with depression was treated with doses of 150 to 450 milligrams per day of Iproniazid for a period varying between 2 and 40 weeks. Some of them were given a placebo. The symptoms of depression were assessed weekly by a registrar who didn't know who got the actual treatment. They found that 26 patients improved, only 12 due to Iproniazid, and the rest due to coincidence. If you were to replicate this study, what questions would you have? For me, the first question would be about treatment assignment. It isn't clear if there were treatment groups at all or how many of them and for how long they were exposed to either the treatment or placebo. Also, there isn't an easy protocol to follow on that treatment. To redesign this, we could have 3 groups of varying doses, 150 milligrams per day, 450 milligrams per day, and placebo. We could also have a dimension on the length of treatment, 2, 4, or 10 weeks. Patients should be randomly assigned to one of the conditions, and neither they nor the people assessing their symptoms should know which group they're in. It's what we call a double-blind trial. You may have realized by now that the limitations in the original study designed by Parry and Sandler compromise our confidence in their results. If we were to replicate their study, there wouldn't be an easy way to compare their findings to ours. It's worth considering that standards were different in 1959, but science operates under the principle that a good study design must be replicable. Thus, papers, and often the accompanying documentation, should exist as the self-containing units of communication so that other people in the scientific community can replicate their designs and further validate or refute their findings. This practice builds trust and credibility in scientific discovery. Perhaps not sufficiently evident in the Parry and Sandler study is that one of their patients died due to acute hepatic necrosis, an abrupt death of liver cells. And I know there is very little we can extract from a very small sample size, but at the same time, if 2% of your patients die during the treatment, that's something that requires further attention. The hepatotoxicity of Iproniazid turned out to be a big problem. In a series of experiments with rats, Nelson, Mitchell, Snodgrass, and Timbrell began to put together the pieces of this network of reactions. Here's what they found. Iproniazid can metabolize into a few compounds, depending on the kind of reaction it undergoes. Through hydrolysis, Iproniazid becomes isopropylhydrazine. With isotope labeling, they figured the cytochrome P450 oxidizes isopropylhydrazine, forming an isopropyl radical. With a closer look at the isopropyl radical, we may pick up on its chemical properties. The big star here is this carbon. When we distribute the electrons of its valency shell, we notice an unpaired electron. This is an unstable state. This carbon is now thirsty to bond and stabilize. But if you remember our discussion earlier about electronegativity, you will know that carbon isn't among the most electronegative elements. Thus, instead of stealing an electron from another species to stabilize its shell, it will form a covalent bond, attaching itself to another structure. This process is called alkylation. If we are to imagine how it interacts with a protein, this radical is likely to attach itself to a negative side of the chain. And because the structure and function are so closely linked, alkylation will ultimately interfere with the function of the protein and end up killing the liver cells in the process. Due to its hepatotoxicity, increased risk of hypertension, and interactions with other drugs, Iproniazid was removed from the market in 1962, only 5 years after its approval by the FDA. But other monoamine oxidase inhibitors are still available to treat depression. Along with tricyclics, monoamine oxidase inhibitors remain the founding class of antidepressants. Okay, time for a recap. Today we learned about the serendipitous discovery of Iproniazid an antibiotic turned antidepressant that inspired excitement for molecular chemists in the 1950s. We reviewed one of the early clinical trials assessing the efficacy of iproniazid in reducing symptoms of depression, noting the limitations in the study design and how to address them. The efficacy of iproniazid strengthened the monoamine hypothesis, which proposed that depression was associated with a deficiency in monoamines. This is when we met monoamine oxidase inhibitors, the class of antidepressants that work by inhibiting the oxidation of monoamines, thus increasing their availability in the brain. Despite its short-lived clinical use, iproniazid inaugurated an era of exciting discovery for psychopharmaceuticals, fundamentally changing the landscape of treatments for depression. But with iproniazid out of the market, who's to step in? Stay tuned for our next video on fluoxetine, America's favorite drug. You can find all the resources used to make this video in the description. There you can also find additional resources that we recommend that are connected to the topics we talked about today, such as a glossary with key terms we defined and an essay on the reproducibility crisis. See you in our next video.